This is the second video on the series of lecture in the cell cycle. In the first video, I briefly give you the overview of the cell cycle and about different phases. If I summarize that, I told you that in the cell cycle, you have the first phase of the cell cycle is the G1 phase, which is also known as the decision-making phase, and it is during this phase that the cell decides whether to go for the cell division or not go for the cell division. If it decides to go for a cell division, it moves toward the S phase, where the DNA that is synthesized, that is replicated for equal division between the two daughter cells at the end of the cell cycle. Then you have got this G2 phase, which is actually the quality control phase. We will discuss that in detail. And then you have got the last phase of the cell cycle, which is known as the M phase, divided into mitosis, which is the division of the nucleus, and the cytokinesis, which is actually the division of the cytoplasm. And in the end, you are going to get two daughter cells after the process of the cytokinesis. I told you that for the execution of the cell cycle, you need a machinery. And the machinery that is made up of different components. The first component that we discussed, that was the centrosome, that was actually working as the microtubule organizing center. And this centrosome that was made up of two centrioles and a mass of protein that was known as the pericentriolar mass. And some of the examples of the protein that were present in the pericentriolar mass, that was the gamma tubulin, the pericentrine, and the ninine. I've told you that the pericentriolar mass, that is actually responsible for a particular process known as the microtubule nucleation, which is actually the event that initiates the de novo formation of the microtubules. And that means you are not elongating already existing microtubules, you are starting their polymerization from the scratch. Today I'll be talking about the process of the microtubule nucleation in a little bit detail, but before going into that, you should understand the second component of the machinery that are known as the microtubules. Now these microtubules, they are the polymer of two proteins. One is the alpha tubulin and the other one is the beta tubulin. So they are actually the polymers of the proteins. Actually, the alpha and the beta, they combine with each other, and it gives you a particular heterodimer. A heterodimer in the sense, hetero referring to different and dimer is referring to two. So if you look at this particular subunit, that is made up of two proteins, but as the two proteins, they are different from each other, we call this as the heterodimer. One heterodimer forms, another heterodimer form, another heterodimer form, and they combine with each other to give this long polymeric structure, which usually refers to the microtubules. These microtubules, they have got two ends. We usually describe them under two ends. This beta tubulin, that actually refers to the plus end of the microtubule. Now this is not the mathematical plus and minus. Here the meaning of the plus and minus, that is a little bit different. Now the plus end of the microtubule, which is actually the beta tubulin, that is that particular end of the microtubule where the process of the elongation as well as the process of the shrinkage that is occurring. That means all of the activity of the elongation and shrinkage that is happening at the beta end of the microtubule. Therefore we consider that as the plus end, because all of the activity that is going on this particular end of the microtubule. Then the alpha tubulin, that is actually referred to as the minus end of the microtubule. That means there will be no elongation, no shrinkage on this particular end, and that is a relatively stable end of the microtubule, providing stability to the long polymers of the microtubules. These microtubules, they are broadly of three types. One that is known as the Kinetic Code microtubule, the other one that is known as the astral microtubule, and the third one that is known as the polar microtubule. We will discuss their function in detail in their respective places. Now, moving back towards the microtubule nucleation process, how that is happening. Now the process of the microtubule that requires the action of a third tubulin, which is called the gamma tubulin, and its associated complexes. You all know that the gamma tubulin, of course, we are talking about the centrosome, because the centrosome, they are made up of the two centrioles and the protein complex, where the gamma tubulin is one of them. Now the gamma tubulin that combined with its associated protein to give you a complex protein structure that is known as the gamma tubulin ring complex. For short, that is known as the gamma TURC. This TU is tubulin, this is the ring, and that is the complex. This is referring to the gamma protein. Now this gamma tubulin ring complex, that is actually acting as a scaffold for the alpha beta tubulin polymerization. And of course, this alpha beta tubulin polymerization refers to the polymerization of the microtubules. It also acts as a cap for the minus end of the microtubule, while the microtubule, they continue their growth at its plus end. As I've told you earlier, that the beta end, the plus end of the microtubule, that is responsible for the elongation and the shrinkage, while the minus end, that is the acting as a stable structure, is a stable end of the microtubule. If you look at this one, like if this is the gamma tubulin ring complex, you can actually see that the alpha subunit that is residing in this gamma tubulin ring complex providing a structural stability, while the beta end, that is exposed for elongation and the shrinkage. So this is one heterodimer, because the beta tubulin that is there, that is going to combine with another heterodimer, and so on, to give you a long polymeric structure of the gamma tubulin. The third component of the machinery, that is the kinetic code. Now the kinetic code, it is a protein structure that is present on the chromatids, where the kinetic code microtubule attach during the cell division to pull the sister chromatids apart. These kinetic code, they are very important structure, because the absence of the kinetic code means that you cannot equally divide the genetic material between the two daughter cells. That is one of the most important protein structure that is present on the chromatids, and especially its function will be visible when we will be discussing the anaphase, where we will be dividing the two daughter cells, the two sister chromatids between the two daughter cells. We will discuss their function in detail over there. If you look at the kinetic code, if this is a sister chromatid, this one is a sister chromatid, this particular structure is the centromere of the DNA, the centromere of the sister chromatids. Do not confuse the centromere with the centrosome. The centrosome are made up of they are an organelle made up of the centrioles and the pericentriolar mass. Centromere, that is part of the DNA. It means you're talking about the sequence of nucleotides. This centromere is interacting with one of the component of the kinetic code, which is called the inner kinetic code. So if you look at the detailed structure of the kinetic code, that is actually made up of two subtypes. One that is known as the inner kinetic code. Now the proteins of the inner kinetic code, they are tightly associated with the centromere DNA. You all know that the DNA is a negatively charged molecule because of its phosphate groups. So those proteins which are actually having a positive charge will be very in good interaction with the DNA. If I give you some of the examples of the proteins that are present in the inner kinetic code, one of the example is the CENPA protein. And this particular protein is actually a variant of the H3 histones, the famous histones that are the small proteins positively charged, which are in interaction with DNA to give them a particular structure. So if you are interacting with the protein with the centromere DNA, it means you're talking about the protein DNA interaction. Another subcomponent of the kinetic code that is called is the outer kinetic code. And this outer kinetochore, it actually interacts with the microtubules, especially the kinetochore microtubules. Now, as the name indicates, that if you're talking about the kinetochore microtubules, these microtubules are going to interact with this kinetochore. Now, one of the major differences between the inner and the outer kinetochore is that in the inner kinetochore, you're actually talking about a protein-DNA interaction. Here we are talking about a protein-protein interaction. If you look at this one over here, you have got the centromere DNA, and the inner kinetochore is in direct interaction with the centromere DNA. If you look at the proteins of the outer kinetochore, on one side they are interacting with the inner kinetochore, and of course the inner kinetochore is a protein structure. If you look at this outer kinetochore on the other side, that is in interaction with the kinetochore microtubules, and of course the kinetochore microtubules, they are made up of proteins, the alpha and the beta tubulin. So the outer kinetochore is having protein-protein interaction, the inner kinetochore is actually having protein-DNA interaction. These are just some one of the example of the proteins that you are going to find in the outer kinetochore, that is known as the BUB1. So if you talk about the machinery, I told you about three things: about the centrosome, about the microtubules, and about the kinetochore. So collectively, sometimes they are called as the spindle apparatus. So in the spindle apparatus, you have got three subcomponents. One is known as the centrosome, the other one is the microtubules, and the third one, they are known as the kinetochore. Another important component of the machinery, that is known as the contractile ring. And this contractile ring, it's play its role in the cytokinesis of the cell cycle, and its function is to pinch the two cells apart from each other. Once you have divided the genetic material, you have done your mitosis, you are going for cytokinesis, and in the cytokinesis, this contractile ring is going to pinch the two cells apart from each other. This contractile ring is actually made up of different proteins, but the most important of them are the actin filaments and the Myosin-II. Now, when you write this 2 with this myosin, this is actually a differentiating term. By that I mean that you have got the myosin protein in the muscles, you have got the myosin protein in the contractile ring. How to differentiate between the two? We put a 2 with the myosin over here. That is actually differentiating that you are talking about the myosin of the contractile ring. You are not talking about the myosin of the muscle cell. So if this is simply myosin, you are talking about the myosin of the muscle. If this is Myosin-II, you are talking about the myosin of the contractile ring. We will continue this in the next video.